let's proceed now with the sequencing data. So I assume here that all of you know Sanger sequencing. And Sanger sequencing is the really old sequencing method. It's a method that people were using maybe 20 years ago, 15-20 years ago. Okay, they use it also nowadays to test things and get some specific regions of the genome, but what you should keep in mind is that really this Sanger sequencing method is not a suitable method for obtaining whole genome data. So this, you cannot really do it with Sanger sequencing, even if they did it actually 20 years ago, more or less. So with Sanger sequencing you can get a sequence of a specific region of the genome and so on, but you cannot get whole genome data. And how it works is really simple. You use a primer, which is here. The primer hybridizes to a specific region on the DNA. So your DNA is this one, the DNA that you want to sequence. You have designed the primer so that it will be complementary and able to hybridize here. So you will have this hybridization here, and then this red part is the part that you really want to sequence. So the primer is known to match this template, this part of the template here, but this one is unknown and you want to find out the exact sequence of this part. Okay, so how you do it is that you synthesize actually this part de novo, but instead of using nucleotides you use ddNTPs. ddNTPs are modified nucleotides, adenine, cytosine, thymine and guanosine, but since they are modified they cannot be elongated. So if such a modified nucleotide will be put somewhere, then nothing else can be put after that. Okay, so how it works is the following. On the top of that you add a different fluorescent label or radioactive label at the end of this ddNTP. So for example the ddTTP, the thymine, has a red label, the CTP has a blue label, the ATP has a green label, and the GTP has a purple label. This is just an example here. Of course in the mixture you put some normal NTPs as well, normal nucleotides. Okay, how this works then is the following. So you have this one ligase, this one hybridizes here, and then you will start to synthesize this part on this sequence. And it's possible that the first thing that will be entered will be a ddNTP, and if this is here adenine, then if it is here a cytosine here, then what will enter here, it will be guanine. So you will have a DT GTP here, and this will light purple. Now, since you have normal NTPs, it's also possible in another primer molecule that a normal guanine will enter. Therefore, you will have this part of the molecule, but then, since it's a normal G, it can be elongated, so then if this is also a C here, another G will enter here, but this G can be D GTP. Okay, then again it will be a purple, but then again you will have another molecule, so you have several millions, or I don't know how many molecules in the liquid here. So it's possible that a normal G will enter instead of this TTP here, so it could be a normal G here. And then if this is adenine, then a thymine will be entered, but this thymine might be the TTP, so it will light red, and so on. So you will get all these different size molecules that you can detect with this in this capillary gel here, and then you can use computer software to eventually get this graph that will tell you what color you read at each position. So this is what we were doing several years ago. This is called a chromatograph. We were viewing images like that over and over again and all the time in order to determine that the sequence of this piece is actually here it's T, G, G, T, and so on. And you can see here that it's not very clear, and of course you might get mistakes, but mistakes you can get everywhere in biology. But the problem with that is that it's a really low throughput. You cannot do this process for the whole genome. It's too expensive and too costly in terms of time. Okay, then after that we were when the next generation sequencing appeared, it used different ideas to speed up the whole process. There are different technologies of NGS. I will give you a paper to study for that. But what we will use really in this class will be Illumina data. And Illumina uses a very similar idea to that, but has enormously managed to speed it up by actually doing several sequencing processes in parallel. So it's like a super optimized Sanger sequencing. That really used amazing ideas to manage to get fast sequences of a whole genome. Okay, let's go now to describe how this works. So how Illumina sequencing.